Today we are going discuss about blotting techniques. Okay, blotting techniques, it is just like a snowdrop. Okay, in conclusion, I will reveal the secret of snowdrop. Okay, first definition of blotting techniques. Blots are techniques for transferring DNA, RNA, and proteins onto a carrier so they can be separated. Okay, and often follows the use of gel electrophoresis technique. Okay, the southern blot is used for transferring DNA. Okay, the northern blot for RNA. Next, western blot for protein. Okay, there are three types of blotting techniques. Okay, first one is southern blotting technique. Okay, next one northern blotting technique. Third one western blotting technique. Okay, fourth one dot plot technique. Okay, this is not important. These three are very important. Okay. Okay, southern blotting detection of DNA. Okay, the northern blotting detection of RNA, the western blotting detection of protein. Okay, next. First one, southern blotting technique. Okay, a southern blot is a laboratory method used to detect specific DNA molecules from among a many other DNA molecules. Okay, the technique was named Southern, Edward Southern. Okay, so the name is called Southern blotting technique. Okay, look at here, step by step guide to Southern blot analysis. Okay, first step DNA digestion. Okay, second step gel electrophoresis method. Okay, third step blotting. Okay, fourth step probe labeling. Fifth step hybridization and washing. Sixth step detection. Okay. Okay, first step DNA digestion. DNA digestion means you take genomic DNA. Okay, next you cut DNA fragments. It can be used restriction enzymes that is called DNA digestion. Next step gel electrophoresis. Okay, gel electrophoresis means electrophoresis carried out on agarose gel to separate DNA fragments. That is called gel electrophoresis. Okay. Next, blotting. Blotting means the transfer of macromolecules by capillary action from a gel to a membrane. Okay, that is called blotting. Next, probe labeling. Okay, what is mean by probe? Probe is a single stranded sequence of DNA or RNA. Okay, used to search for its complementary sequence in a sample genome. Okay, the probe is labeled with radioactive or chemical tag that allow its binding to be visualized. Okay, that is called probe labeling. Next, hybridization and washing. Okay, are you understand? Hybridization at the molecular level. Okay, it refers to joining together of artificially separated nucleic acid strands. Okay, via hydrogen bonding between complementary bases. Okay, it is called hybridization. Okay, next detection. Okay, last step detection means uh, detection of DNA, that is called detection. Okay, your result. Okay, that is called isolation of DNA, detection of DNA. Okay, look at here. First step, southern blotting technique. Okay, isolation of DNA. Okay, this is genomic DNA. These genomic DNA cut the DNA fragments. Okay, it can be used restriction endonuclease enzymes. Okay, look at here. First step, isolation of DNA from nucleotide cells as from the scene of the crime. Okay, first, the desired DNA is isolated from the whole DNA present inside the nucleus. Okay, that is called genomic DNA. Whole DNA means genomic DNA. Next step, okay, cut the DNA fragments. Look at here. Cut the DNA fragments. It can be used restriction endonuclease enzymes. Okay, after cut the DNA fragments, incubate specimen with detergent to promote cell lysis. Okay, now you understand? These lysis free cellular protein from DNA. Okay, look at here. This is DNA fragments. Okay, next step, agarose gel electrophoresis technique. Agarose gel electrophoresis refers to the movement of charged molecules in an electric fields. The negatively charged molecule movement towards the positive electrode, while the positively charged molecule migrate towards the negative electrodes. Okay, agarose gel electrophoresis is a routinely used analytical technique for the separation, purification of specific DNA fragment. Okay, the gel is composed of polyacrylamide or agarose. Okay, it can be used agarose. Okay, agarose is a polysaccharide. Okay, the DNA samples are placed into the wells of the gel surface and the power supply is switched on. Okay, as the DNA is negatively charged. DNA fragments, move through the gel towards the positive electrode. Okay, the rate of migration of DNA is dependent on the size and shape. Okay, in general, smaller linear fragments move faster than the larger ones. Okay, hence gel electrophoresis can be used for the separation of a mixture of DNA fragments based on their size. Okay, look at here. This is l- low molecular weight. This is high molecular weight DNA fragments. These are DNA fragments. Okay, this is high molecular weight DNA fragments. This is low molecular DNA fragments. Okay.